studying people, but anyway. Yeah, so this is a topic that I got into because I thought, how can you do a paper quickly? Meta-analysis might be quick. And I realized that's not the case at all. After doing quite a lot of work already. Anyway, and as you'll see from this, there's quite a lot more to a meta-analysis than initially meets the eye. So why would you want to do a meta-analysis? So individual experiments often rely on really small sample sizes just because of the constraints of time and manpower and money, etc. Get a whole bunch of studies to increase your statistical power. And then studies on the same topic often differ in their experimental methodology. So they might use different species or different analytical approach, and then they often lead to very different conclusions. And it's difficult to evaluate that just on the face of it without some sort of quantitative method. So meta-analysis allows us to do two things. So if the number of attempts have been made to measure the effect of one variable on another, then meta-analysis provides a method to calculate the mean effect of the independent variable across all of these attempts. And then secondly, you can measure the change in that dependent variable across studies and attempt to explain this variability using moderator variables, which we'll talk about later, which are basically just predictor variables, but they're called moderators for whatever reason in meta-analysis. So the big thing with meta-analyses is the quality of doing them. And you'll see the literature talks a lot about how you do these things, and there's actually quite strict guidelines now for how you do them. Because the conclusions can be very dependent on actually how you go about doing meta-analysis. And biological meta-analyses in particular have been singled out for being rather poor in the way they do things. Compared to, say, medical sciences, where they're very strict about these things for obvious reasons. So I thought to make it a little more light-hearted, ask you the question, what do anime and human sexual behavior have to do with meta-analysis and our result for today? And I don't know if anyone knows where this comes from. Any guesses? Yes. So I didn't know that. Anyway, the reason I have that there is because this man's favorite movie, not just anime movie, is Totoro. And he was doing a PhD, he was a few years ahead of me when I started my PhD. He was almost finished actually. Really brilliant Japanese guy. He published 10 papers out of his PhD. But a really, really cool guy also. And he's written quite a lot about meta-analyses. And I'm going to be basing this talk largely on this particular paper. But at the end, I'll give you references to refer to. And where sexual behavior falls into the story is at Sheffield University, where I was. They had a human ecology research group, and Shinichi worked with them, although that wasn't exactly his main focus. But they did quite a lot of work on looking at human behavior. So it's quite interesting for an ecological group to actually look at humans, so from like an animal perspective. And I'm going to use a study that Shinichi was a co-author on as sort of an example to explain some of the things about meta-analysis. And there's this thing called make choice copying. So it's been seen a lot in animals. Actually, they've looked at it in humans quite a lot. So make choice copying is it's the probability of acceptance of a potential mate increases if one sees this person being chosen by others first. And that's basically what these photographs show. And what they do in like these tests is they show people photographs like that. So you have to rate the attractiveness of these people. So either by themselves or surrounded by a bunch of guys. Yeah, and so I've got this little cartoon here which you see throughout the talk, indicating that it's referring to this paper. And then I've divided it up into different sections. So this is just the meta-analysis process that you need to go through. I'm not going to read through this now, but I've kind of color-coded it so you'll see that's why I've got these weird colors in the background. And just to show you that there is quite a bit involved in the meta-analysis process. So the third thing, obviously, is the literature search. And one of the things that Shinichi emphasized in their paper is that it needs to be systematic. So it's critically important that it needs to be systematic. So you need some sort of predetermined protocol for minimizing bias in the resulting data set. And there's this thing called PRISMA, the preferred reporting items for systematic reviews and meta-analyses. I don't know why they chose that name. Sounds like it was chosen to suit the acronym rather than the other way around. And this is a very big thing in the health sciences, but you'll see that it's being applied more and more in the biological sciences, and it's actually required in many journals. And I'll speak a bit more about that in detail. And you can refer to this paper here for a detailed description of PRISMA. So I'm just going to skim over what PRISMA is about. And one of the major things is this, what you see on the screen there, which is called a flow diagram. And basically you're meant to show all the steps you went through in your literature search. So where you search and how you basically remove studies as you go along. So I'm going to go through all the different steps. So the initial step is the data search. And this should be as comprehensive as possible. So Shinichi then say that you obviously should try and be as comprehensive as possible, but being systematic is far more important than being comprehensive. So they suggest that you use at least 2 major databases, so Web of Science, Scopus, Google Scholar. And the reason for this is, as they say in the paper, and this is another paper that they refer to, the overlap between these databases is actually quite low, surprisingly enough. So this is natural sciences and engineering at the top, and you can see that the overlap there is something like 45% between Scopus and Web of Science. So it really is quite low. And then also using other sources of published and unpublished studies. And you'll see they go into a lot of detail in some books and papers about what you can do. So for example, using backward and forward citations, emailing people for unpublished data, etc., etc. Okay, so in our make-choice copying paper, you can see that they really gave a lot of detail about what they did. I'm not going to read all of this to you. It's just to give you an idea of the sort of detail that they are looking for in a PRISMA type process. Okay, so then the next step is screening and eligibility. So you've now got your thousands of papers I found. And now you need to try and whittle away at those to find studies you're really interested in. So what you need to then say, the criteria might differ for the initial screening, where you're just looking at the titles and abstracts, with the full text screening, but you need to report both in detail. And the criteria examples can include, it needs to have certain experimental design requirements. Some of the papers don't always report the outcomes, or they do, but it's not exactly as needed. We'll go into a bit more detail about what you actually need. So they might miss things like variance and sample size, which you actually need to calculate certain things for a meta-analysis. And then also you might get duplicate results, so two different papers actually report on the same sort of thing, or you get, or what I also found when I was doing this is you get these long-term studies, and actually you only need the most recent one because they report all the results in there. So in R, there's a package called Meta that allows you to do abstract screening. I didn't use that, but it is available if you like it. And then another one that's quite big apparently in the health sciences is this standalone package called Abstracto. And it's apparently like a machine learning, it has a machine learning algorithm that will actually search the abstracts for you, and it's supposed to be reasonably good. So I recommend that you should have at least 2 people in place for this process, and then some sort of plan in place for if there's disagreements, and then reporting disagreement rates. Yeah, I must admit I haven't seen people reporting sort of thing in meta-analysis papers, but yeah. And then the list of studies excluded and the reasons should also be reported, and you'll see that in the example here. So in our matrix copying paper, they excluded 23 papers. So some of them were reviews, there was no data in them. Some of them were just a theoretical framework. Some of them were mathematical models. And then 69 articles that were not focused on humans were also excluded. And then they gave very detailed information about their including and excluding criteria in the paper. And I'm not going to read those to you. Okay. Then, once you've got your final data set, you need to now start extracting data for them to calculate your effect sizes. And again, I recommend you should have at least 2 people. You should say from where in the paper you get it. So this will obviously all go in the supplementary material at some point. And then reporting intermediate calculations, transformations, etc., etc. And obviously that again will probably go into your supplementary material. When I was doing my meta-analysis, as I'm still doing it, I used there's an online thing called Webplot Digitizer, and you can also download it, that allows you to take a figure in and you can click on it to get the data points. And there's a bunch of other ones that I haven't tried out myself, but I also recommend it. Obviously, if you can get the data from tables in the paper, that's better. And then you can also email the author sometime. You might be lucky and get the data that way. Okay, and then the final step in the Prisma process is what they call documentation, and that's usually the production of this flow diagram. So this is from a matrix copying paper. Unfortunately, in paper, the resolution of the graphics was quite terrible, so this wasn't because of my copying, it was bad already. But anyway, you'll see that they give the sample sizes at each step, and then the mean potential other things. And then in R, there's a package called meta-analyses that actually allows you to do these prisoner flow diagrams. In the Shinichi's paper, they also recommend that you state the software that you use. I think that's pretty standard practice. And then also including the code for the analysis in addition to the data. Although I'm only seeing that done in the papers. Or meta-analyses I looked at either. Okay, so the next step in the meta-analysis process is deciding what the question is and what effect size fits that question. So all good meta-analyses will ask a question and test a hypothesis, and the specific effect size will depend on the specific question it is. And I'm going to talk about the three main types of effect sizes, but please note that there are many more. And if you look in the references that I've given you, you can find more details about them. And there's also all sorts of things, depending on non-independence in the data, you might use different calculations, etc., etc. Okay, so the first type of effect size are when you're looking at differences between the means of two groups. And often this is for experimental studies, so you have a control and a treatment, and you get the mean values from those two to calculate your effect size. And most commonly used effect size statistic for these types of studies is the standardized mean difference, and you'll see that this they refer to D currency, wedges D, wedges G, and they're all basically the same thing, just sort of slight variations around the calculation. And this is just the formula for currency G, so it's very basic. It's just the mean of the one subtracted from the other, and then divided by the pooled standard deviation. And the formula for that is down here. So the pooled standard deviation, if you imagine with the two studies, or not two studies, sorry, your two treatments, each one will have an effect, a standard deviation associated with it. And now you calculate them one value from these, so you need sort of one measure of variability, and that's basically what this pooled standard deviation is doing. The other thing to remember with these effect sizes is that they are unitless. So if you have a study, for example, trying to think of a good example. So if you have one study that's looking at body weight, and then another one that's looking at body length, the units there are different. So one might be grams and other centimeters or whatever. But when you calculate the effect size, because it becomes unitless, you can actually compare the two. And that's the cool thing about meta-analysis. This is just sort of guideline values on what a small, medium, and large effect size might be. So 0.2, 0.5, 0.8 for all these values. And I think this was from Cohen's paper way back in the 80s. Okay, the second effect size statistic you might use for the differences between the two groups is the natural logarithm of the response ratio. You can see that it's very similar to standardized mean difference. But here you're just dividing the treatment by the control and taking the natural logarithm of that. And then there's an associated measure of variability with that. Okay. The next type of effect size statistics are looking at the relationship or correlation between two variables. So this is often for observational studies. And there's a thing called the Fisher's Z transform correlation coefficients. And you see here R is for correlation coefficient in this formula. And again, there's sort of guideline measures of small, medium, and large effects. And then the last type of effect size measures are the incidence of two outcomes, and often these are reported in contingency tables, and you would calculate it from a contingency table. So, for example, dead or alive. So you have your control and your treatment, and then that two different outcomes, so whether they're alive or dead. And there's two major statistics associated with these, so the odds ratio and relative risk. So you'll see here it's also the natural logarithm RR, so don't confuse that with the response ratio in the earlier one. I don't know why they gave it the same notation. And you can see here the ABCD is just referring to the different parts of the contingency table. Okay, so something to note is that a lot of these effect size statistics are actually interconvertible. So, for example, if you have experimental studies and observational studies and you've calculated the Fisher's Z transform correlation coefficient for observational studies and the standardizing difference, you can actually then get them all to the same statistic, which is really nice. So it's just expanding your ability to look at a wider range of studies. And I haven't provided the formula for these, you can look them up. Also just wanted to point out that there are other effect size statistics that could be useful for biologists. You could look at survival curves. You could also look at the differences between variances rather than means. So you might be interested in how wide a particular variable is rather than the mean. And then these two papers provide details and formulae for a lot of these things. In R, these three packages are probably the most commonly used. But you see later on, I give a link to a website for that. It's one of the CRAN special topics. And you'll see there's actually a lot of packages that you can use. Okay, so in our example, the tests that they investigated, so they were interested in what they call addition, so of two subjects alone versus with an opposite sex. So that's kind of what this figure is showing here. So you have a person by themselves, and then you add guys around the girl, and you measure the attractiveness of the person, or whatever the measure is there. Or what they call augmentation. So this is a rather crueler one. They put a person with another person of the opposite sex who's unattractive, and then someone who's attractive. So I made a subjective call, and he was attractive and unattractive. And then they looked at that for males and females. So they had 4 different tests there. And then they used the natural logarithm of the response ratio, because experimental designs differed amongst the studies that they had, and then the units of measurements also differed. The other reason I said they used this was because it doesn't depend on sampling standard deviation. So you'll remember standardizing mean difference, you need to calculate the pooled standard deviation, or it's calculated, whereas this doesn't actually need that. And I said that was problematic because the way that these studies are done, if you give a test subject a picture of a person, so say a girl, and then a girl with one guy, or a girl with two guys, three guys, etc., just that alone can actually increase the variance. And this is just an example of a paper in this matrix copying paper that they got the data from. So you can see I went and I pulled the figure out, and then they obviously would have used one of those software packages and got the values there, and then calculate the effect size using those formula, this formula here, and then the variance there. Okay. So one of the big things we have to worry about in meta-analysis is non-independence. And Shinichi will talk about 4 major sources of non-independence. Although I think there's actually a 5th, which they don't mention, but which I will mention. So in this figure here, you can see here you have your different studies, and then they looked at control and treatment type setup. You know, it doesn't have to be like that. And then ES is effect size, and then this little arrow here is showing the correlation between the effect sizes, so the non-independent. So in the first example, you could calculate multiple effect sizes from the same study, and that's obviously not independent. In the second example, this is when you have shared measurement. So this could be, for example, what I mentioned earlier, but within one study, so you've got 2 different traits, so like body length and body mass, which are proxy of body size, and you could actually calculate 2 effect sizes from the same paper, but then those are not independent. A third major source of non-independence is a shared control group. So often in experimental setups, you might have, for example, like a heating treatment. So when you increase by 10 degrees or 20 degrees, but then your control is still the same. So treatment 1, treatment 2 are using the same control group. And then the fourth one that is quite unique to biology is phylogenetic non-independence. I'm sure all of you will have come across this in other contexts. So it's just due to shared evolution in history. And there are ways of accounting for biology in meta-analysis. But the fifth source of non-independence that's not mentioned here is spatial correlation. So like a lot of what I've been looking at, for example, like productivity, you could imagine when you have studies, if studies are close to each other, that it could be correlated just by virtue of the fact that they're close together. I'm not actually sure if anyone has tried to control for that in a meta-analysis. They probably should. Okay, so how do we control for these different sources of non-independence? We use different types of models. And there's 3 major types of meta-analytic models. So fixed effect models, random effect models, and multi-level models. For fixed effects and random effects, so there's 1 effect per study, and assuming there's no other sources of non-independence. So we're assuming that all effect sizes are independent for those two models, whereas the multi-level model relaxes those assumptions. But I'll go into detail about all of these now. Okay, so the fixed effect model is the most basic of these, and it assumes that all effects share a common mean, which is what this figure here is trying to show, but I'll talk about that in the next slide. So variation among effects is solely due to sampling error. And that's usually not realistic, especially in a biological type study. But it can be justified if the effects are all from the same population or species, and assuming that the effects are independent. So you can imagine that it's quite an ask actually to justify using a fixed effect model. So this is what a typical fixed effect model looks like. So yi is the observed effect for the I study. So I'll show you now. E naught is the overall effect of the mean, so that's this line over here, and that's the effect that we are interested, that we're trying to calculate. And then EI is the deviation from E naught for the art study, and that's what these little orange lines here are showing. Okay, so for each study, you're calculating, you're trying to estimate with these things in order to calculate the mean effect. So the random effects model relaxes the assumption that all studies are from the same population. So, for example, different study designs are likely to have different effects. So how you went about doing the study can actually influence the effects. So the random effects model allows you to account for different studies. The problem though is, and this is a guideline that they have in the paper, they didn't reference it at all, but they say that you probably need more than 10 effect sizes to estimate between study variance. So for that reason, if you have fewer than 10, you might want to go for a fixed effect model, even though it's probably not correct. This is what the formula for a typical random effects model. So the same as for the fixed effect model, except we have this study-specific deviance, SI. And SI is distributed with the variance of tau squared. So that's your between study variance. So what this is showing over here. And that is what these green curves are showing here, between study variance. So you still have your same as for the fixed effect model. You have the deviation for each study, so in orange, but then you're also looking at a study specific. Between study variance. Okay, and then lastly you have the multi-level model. And this allows for multiple effect sizes from the same study. So for example, different experimental treatments, but you could also have other levels. So in addition to different experimental treatments, you could have a species level on top of that. And it could get as complicated as you wanted it to be. And the multi-level model adds on this Uij, which is the deviation from SI for the J effect size for the I study. So that's assuming you're adding this study adding on a study level to this multi-level model. Okay. Okay, and you'll see here we have this additional blue curve here, which is the within study deviation that you're now estimating. Okay, so in our main choice copying example, what they did was they assumed that there was non-zero between study variance. So, in other words, they were not independent. And so they used a random effects meta-analytic model for 1 to 3. So that was the addition for males and females, and then augmentation for females. But then for model 4, which was the augmentation for males, if they didn't have enough samples, they did a fixed effects model. Okay, so that's just an example of what I was talking about here, the difference between the random effects and fixed effects model. Okay, so once you've calculated your effect sizes and you run your meta-analytic model, typically you would display your results in what are called forest plots. That's what these are showing here. And depending on your effect size measure and whether, say for example, you were looking at phylogenetic non-independence, you can report them in different ways. So you'll see here the little dot or square or whatever will be the effect size for each study in this case, and then the overall effect size there. And then this is for a fixed effect model and a random effects model here. And then there's a confidence interval associated with these. The other thing I want to point out is often they provide the number of effect sizes associated with each observation over here. So they got 17, 15, etc., etc. So this is probably a better example. So this will be for different species. You've got different numbers of effect sizes, and here you've got different treatments or different genders, etc. And in this one here, when you're looking at the correlation coefficient, what they've done is they've color-coded it by those guideline strength of the effect size. So a strong effect size is about 0.5 or whatever. Okay, so in the main choice copying study, and this is, I guess, the interesting part of the paper, what they found was that so this is for the addition studies and this is for the augmentation studies. In the addition studies, so for female choice, so this is where men are choosing females, there was a positive effect. But for male choice, there wasn't. You can see there's a line there, it goes over the zero line, got an overall effect. And then for augmentation, on the other hand, they found positive effects for both female choice and male choice. So, yeah, it's quite interesting, I thought. And this is, I just highlighted this because this is the example where I calculated the effect size and the variance. Okay, so once you've done all of that, what's really important is to report heterogeneity or inconsistency. Okay, and the reason for this is that you could get a mean effect size of 0 when all your different effect sizes equal 0. So I kind of tweaked that earlier figure to show that here. So you can see each study is actually on the 0 line, so there's no variation around those studies. And then your between study variances there. So there is variation, but the between study variation is 0. But in this example, you have very different effect sizes, but they're centered on 0. So you put between study variances greater than 0. And the reason why we worry about this is because it could be an indication of publication bias, or there's other factors actually affecting it that you haven't taken into account. So one of the measures of heterogeneity is Cochrane's Q. I'm not going to go into detail about it because I don't see that many people report on it. But it's one of the recommended ones. The other one is the heterogeneity statistic, this R squared. And it addresses some of the issues of Q, and it ranges from 0 to 1 or 0 to 100%. And it indicates the percent of the variation in effect sizes that's due to between study variance. And there's also some recommended sizes. But what Shinichi notes is that in biological sciences, heterogeneity is often very high, so these guidelines might actually not be that useful. And you can actually see in the macroscopic paper, they were generally quite high, although this was the only one that was significant. So as I said, that heterogeneity could be due to other factors other than sampling variation, and these are your predictors in the other type of model. So, for example, in the macroscopic paper, they recorded whether the people were young or old, high or low fertility, etc., etc. But because the sample sizes were too low, they didn't actually do any sort of meta-regression. Something to note when we are doing meta-regression is that the same rules apply as for multiple regression or regression in general. So you need to think about statistical power. So will you be able to detect an effect if you have lots of moderators but not many effect sizes? And then multicollinearity. So is there correlation between the moderator predictor variables? Okay, and then Shinichi then recommends doing a bunch of sensitivity analyses. So one of these is publication bias, and one way of looking at it is these funnel plots. And basically you just plot all your effect sizes against the size of the study or the variance of the study. And what this will tell you is if you are missing studies. So this is an example of a unbiased meta-analysis. But in this one over here, so the black dots represent significant results or significant effect sizes, and these are non-significant effect sizes. Sorry, not effect sizes, non-significant results in the study, and these are significant results in the study. So often you might imagine that non-significant results are not reported. So if your final plot just came out like with all these black studies, you definitely know that there's a publication bias to just significant results. Also, what often tends to happen is studies with small sample sizes are missing. And again, here, even if they did have all this, the non-significant results, these black points here falling out of the 0.95% confidence intervals here indicate that it's biased towards studies with a positive effect size. Okay, and there's ways of trying to calculate the number of studies that you're missing. So this is what this trim and fall analysis does, and which is what they use. I'm not going to go into detail about that. And then there's a bunch of other sensitivity analyses that they say that you can try and do. So you could try and exclude studies that have a very large effect size, and that might be biasing the results. And then missing data, this apparently is quite a new area in meta-analysis, that using data imputation to try and fill missing, for example, variance data or moderator data. Okay, so in R, within meta-analysis, probably the two most used packages are meta-4 and meta-gear. There's also Bayesian approaches available. But you can check this website out for a list of all the different packages. There are a lot. And when we upload this talk, I'll give a very small example using meta-4. And then finally, the bigger picture of meta-analysis is that you're supposed to be generating review-generated evidence as opposed to study-generated evidence. So you need to discuss what has been confirmed or refuted and what the new insights are from your meta-analysis. And then these sort of things are what you typically associate with review papers, but which equally apply to meta-analysis, is identifying knowledge gaps and then looking at how the type of work that is needed to move the field forward. Okay, and these are the references I mentioned that I think are quite useful. This book is really good if you want to go into detail. And that's it.
Yes,